Good afternoon to everybody. My name is Carlos Sala, and I'm Health and Nutrition Sales Manager of Sacco System. Today, the Sacco webinar will be discussing about the targeting the brain-gut microbiome axis by ImmuneBiotech, a new therapeutic avenue for management of neurological disorder. And it's a great pleasure to host my friend Sharam Lavazani here in connection. But let me explain just a bit myself. I'm Carlos Sala. I have a degree in Science and Technology of Food, and I joined Sacco in the 2012 for the first 6 years in the R&D department and developing several strains in the pilot plant of Sacco. And in the last 2 years, I joined the Health and Nutrition Division as a technical sales manager, following directly Asia market, several countries in Europe, and all the Latin American countries. Sacco System is a network of 4 different companies that are linked together to support formally food, culture, and life. Calificio Clerici, Centro Sperimentale del Latte, Sacco, and Chemicalia. All these companies are following 7 main values. One of them is the family company. In fact, we have a family that owns all these companies, and now we have the 5th generation of this. We have also good research inside. We develop a lot of studies. We are investing in collaboration with university and research center, and we invest 6% of our turnover in R&D. What is important is also underline the number of production plants. We have 7 production plants and more than 360 collaborators that are working in our companies. And what is important is also underline the different divisions that we have. We have different business units, starting from culture and probiotic, that is divided in dairy culture, health and nutrition culture, agroveterinary culture, and meat and other foods culture. Enzyme, additive and coating, and labware, that are equipments for all the lab around the world. And now it's a great pleasure to introduce Dr. Sharam Lavazani from ImmuneBiotech, founder and CEO of ImmuneBiotech.
Thank you so much, Carlo. Good evening, everybody. It's wonderful to meet online. I first of all would like to thank you, the webinar team, and also Sacco System for this kind invitation. I would like to first introduce a little bit myself and the company, which is located in Lund, southern Sweden, which is also famous for its university, where I have been affiliated as an associate professor in teaching immunology for almost 20 years. This takes only a fraction of my time these days, but still forcing me to the edge to keep myself updated on this wonderful, exciting field. Thanks to the success of our research, we have started ImmuneBiotech, which initially received great support from the university, protecting the ideas and the product. And the company has also been a member of the Smile Life Science Incubator, where we have received a great support and coaching, translating our research to business. The company is physically located at Medicum Village, which is a wonderful science and business park in Lund. I think we all agree that we are in the middle of microbiome revolution. We are exposed to increased amount of the information connecting the disruption of our gut microbiota and microbial community to almost all the known human diseases. We also start to understand the impact of these microbes on our drugs, the efficacy of our drugs, and also on other extraintestinal tissues, creating scientific fields like gut-brain axis, gut-lung axis, and etc. It's a very exciting, but at the same time very young field, and requires clear understanding and also increasing the healthcare innovations. Within our company, we discover, develop, and since October last year, commercialized therapeutic effective probiotic products designed with pharmaceutical precision for management of gastrointestinal as well as autoimmune conditions. In my presentation, I'm gonna further describe our collection of 150 probiotic lactobacilli and our screening platform in order to design effective products for a purpose and condition, not by random. I will further describe and share the translational success story of our first product, GutMagnific. And finally, I will present the pipeline of our companion probiotics, where we produce and develop and design products to be used to synergize the efficacy of the drugs, as very selected drugs for treatment of autoimmune conditions. There are increasing evidence on alterations of the gut microbiota in autoimmune diseases. The impact of the environment, diet, infections, and the use of antibiotics have been broadly discussed, but it's not really clear whether these observations of the dysbiosis is a cause or a consequence of the disease. So, go to my next slide here. Yes. My research has initially been started in multiple sclerosis, which has originally been recognized as an inflammation of the central nervous system. So basically, there are tremendous amount of evidence showing that during the development of the disease, there will be an increased infiltration of the inflammatory cells and release of a tremendous amount of inflammatory and molecules and cytokines, creating an autoreactive scenario, attacking the myelin of the neurons and disrupting the neurons' signal transmissions. But recently, there are increasing amount of evidence showing the involvement or pathological changes of the gut and also the dysbiosis. So I have the great pleasure being among of the first research groups in the world providing scientific evidence on the changes of the intestinal barrier function during the development of the MS. So these observations have been shown both in animal models as well as the human. But on this slide, I would in particular invite you for a notice on the increased amount of the akkermansia being observed in this disease, where akkermansia have in other neurological diseases has been considered as a good guys or good bugs being reduced during development of other diseases. So by just simply replacing a bacteria that is causing problem was reduced is really needs and requires careful studies where perhaps these could cause other circumstances. Using the same model for MS, where we basically immunize the ordinary C57 black 6 animals with a peptide from the MOG protein, myelin oligodendrocyte glycoprotein, where the animals after a few days start to show symptoms of paralysis, which we just give different scores depending on the development of the symptoms. This is in particular a wonderful model for multiple sclerosis, showing the infiltration of the autoreactive T cells attacking the neurons and causing this symptom similar to MS, but at the same time is one of the best chronic inflammatory models in the world, where once the disease starts, it will be prolonged for a long period of time, giving the opportunity to study the different mechanism of chronic inflammation and, of course, testing different interventions. In this animal model, we have shown that on top of the classical observation of the inflammation, where you can see in the slides by the brown dots on sections of the brain and spinal cord indicating the inflammatory T cells, we have also shown pathological changes or issues in the gut of these animals, and in particular an increased intestinal permeability. And the most interesting finding was that these changes in the gut starts, as you can see here, at day 7, which is before the symptoms of the disease are being observed. So this is quite interesting, and there are other ways to induce the disease by actively transferring or adaptively transferring MOG-reactive T cells intravenously and inducing the disease. So doing the same protocol by just injecting 10 millions of autoreactive T cells or MOG-reactive T cells, we have observed these changes of the intestinal permeability even stronger. So that was quite interesting observations, which gives us the reason to put several years of research by investigating different segments of the small intestine, like duodenum, jejunum, and ileum, as well as the colon, and showing different pathological changes very much equal to what has previously been observed in inflammatory bowel diseases, IBD, like Crohn's diseases. So we have seen Crohn's disease in MS by showing different changes of the structures of the intestine, increased amount of inflammatory cells, both regarding the adaptive as well as innate immunity. And we have also seen increased amount of goblet cells and infiltration of neutrophils in the colon. So it was quite interesting findings, but at the same time, that was very clear evidence on that there is gut pathology and dysbiosis were in fact consequences of a CNS inflammation. So we believe that once, perhaps our hypothesis is that in autoimmune diseases, the disease can be initiated by unknown reasons, nobody knows, but once the gut will be involved, perhaps the acute inflammation will further develop toward a chronic inflammation, and it will be much more difficult for the body to get rid of and manage the disease. We are very much interested on providing a therapy or treatment. So that has been the focus of my research for years. So by using the classical way, screening protocols, so I have been interested to evaluate the impact of the probiotic lactobacilli for many years ago by simply starting a prophylactic treatment, preventive protocol by giving different kinds of probiotic lactobacilli and by mixing in the flask, and they've been drinking the animals 2 weeks before the immunization and also every day after immunization, freshly provided bacteria. And to our surprise, we have actually seen that some of the strains, not all, could delay the onset and also result to a suppressive development of the disease. So, of course, human scenario is not the same. A lot of things works perfectly in the prophylactic and protocols in these animal models. The challenge is a therapeutic approach. So we have a very tough protocol by inducing the disease and waiting 2 weeks after the onset and test a therapeutic product or approaches. So this is very even seldom that even the drugs available today in the market has been really much effective in these protocols. But anyway, that's our interest, the challenge, and we have seen that none of those strains that work perfectly in a prophylactic treatment did work in a therapeutic approach. So not giving up there, but instead inspired by research by that time, so we started to understand the impact of any strain and also combining them, and finally achieving a therapeutic effect by a certain combination of the bacteria. And that was a wonderful scene, since the animals here, they are highly and severely paralyzed, and just recovering from this paralysis, jumping on the floor and running in the cages has been, every time that we repeated the experiments, a wonderful view. So what we learned was that a therapeutic product is for sure increasing activation of the anti-inflammatory mechanism in the gut and the gut-associated tissues, in particular, increasing amount of the anti-inflammatory regulatory T cells. But this has not been has previously been shown in even using other probiotic bacteria. But to our interest, we have seen this increasing amount of cells not only in the gut and the gut-associated tissues, but also in the periphery, in the spleen, and even in the CNS. But we do not believe that the bacteria that has been administered orally, ending up in the gastrointestinal tract, are really reaching out to the CNS, but rather triggering a mechanism that can favorize the activation of the anti-inflammatory cell populations. So in that particular reason, we have started to analyze how each probiotic strain can contribute to this achievement. And we actually have learned a little bit of the immunological mathematics, where and this is very much based on the complementary studies. There's no space showing all this data, but we have shown that IL-10 is very much important for this therapeutic efficacy, where IL-10 knockout mice completely lack of the potential for suppressing of the disease using this probiotic product. But IL-10 has been triggered in the serum by only certain strains, not all. But we have seen that other strains could increase the serum levels of other cytokines like TGF-beta or IL-27. Then together, these could in fact synergize the activity of IL-10 released by other cells. So basically, each bacteria are contributing somehow to some pieces of the puzzle for the whole picture, and reaching out to systemic efficacy. So, inspired by our data and results, and learning from others, we understood that in order to find the ultimate solution targeting the gut, just by replacing or fixing the causes of the dysbiosis would not have a strong clinical efficacy. So, and for that, we have decided to design products with a multi-targeting impact, and for sure we want to fix what is causing dysbiosis. But on the other hand, these bacteria have a major and live interactions with the intestinal barrier function, all the epithelial cells, tight junctions, and also the immune cells, our immune system. The majority of immune cells are actually in that area, mucosal area. So our products are aiming to have an impact on the dysbiosis known for a particular condition, and at the same time restoring and having an impact on the intestinal barrier function, as well as a major impact on the desired immunomodulatory mechanism needed for that disease. So for this, we have set up the approaches with the pharmaceutical precision, and thereby created a complete new category of probiotic products called ImmuneBiotics. In order to develop ImmuneBiotics, you need tools. And of course, the first and best tools is to have access to a variable bacterial collections or library to choose the properties of interest. And this is something has taken the first 3 or 4 years of our time in the company by collecting complete new probiotic lactobacilli from food and healthy food. So what is considered healthy for a human? So I think those of you who have been working with this area, you know that how much work it requires by isolation and characterization. So we have done on each single, by isolation of the 400 clones, we have ended up on 150 unique lactobacillus strains. All of our strains are happily on the EFSA's QPS list, considered safe, no spore formation, sensitive at least to one antibiotics just in case, and simple to culture, facultative anaerobic, which has been very much appreciated and confirmed from our collaborator, Sako, in producing our first product in a large industrial scale. At the same time, we have developed screening platform with different arms and different focuses. The dysbiosis arm, of course, we are interested to see if we can suppress and inhibit the growth of the non-wanted bacterial strains, pathogens or pathobiomes causing the issues and problems in dysbiosis. The fermentation profile and microbiological properties and immunological properties we have on each single strain gives us the tremendous opportunity to start a screening with the good candidates. So we are not really screening 150 strains for any purpose or property of interest. Colonization, of course, is important. In some certain points, it is important to understand whether they do colonize or not. But although it is now completely accepted that the colonization not necessarily guarantee, but the bacteria could actually have an impact on the health even not colonizing. Immunomodulation, as an immunologist, I have been very much interested to analyze the immunological mechanism of this strain. We have developed internal ex vivo models and also in vivo the models with the very relevant assays. I have been criticizing the other previous works by studying the interactions of the bacteria to the human immune cells isolated from blood, because as an immunologist, you know that the phenotype of the immune cells in the periphery is quite different than what we have in the mucosal area. So it's very important to understand the mechanism on the relevant immune cells. So we have technology giving us the advantage to measure different phenotypes of the immune cells triggered by different bacteria, and also to measure cytokines and also measure the B-cell activations and different antibodies. Intestinal permeability is for sure is very important for us, using the same model, but translating it internally with a special protocol, make it suitable for screening purposes. We have a model with a spontaneous increase in permeability as well as a peripheral inflammation, which is as close as you can come for a human scenario, taking a large distance from the other IBD models where the permeability changes is very much caused by using chemicals damaging the intestine, creating inflammation, etc. So this is an example and scenario of the screening for the ability of our strains for activation of the anti-inflammatory FOXP3 expressing regulatory T cells. So as you can see, different probiotic lactobacilli, although you can call all of them lactobacilli, they are so different in that property. And these assays are not only permitting us to check for the property of our own strains, but also to compare to the other commercially available strains. This is a scenario for screening against the Clostridium difficile. We have received a question and request from clinicians at the infection clinic at Malmö University Hospital if we could provide a product for patients they had with the recurrent Clostridium difficile infection. We have asked actually they send us all the pathogens or pathobionts isolated from the patients in the freezer, and we have received several, including to NAP1027, if you might be aware about. These are the hypervirulent strains, has been associated to the death cases in Sweden and also other countries in the world. So we have performed a tremendous screening, and as you can see, the plus means the inhibition of the growth of the strain, tested strain, and the minus is no impact, no effect. So it is so different, and by this we could have opportunity to finally come up with the combinations with the best interest inhibiting the most or all. This is another way of screening. We have been extremely happy with the collaboration with the research group at Lund University, checking and screening the ability of our strains for production of short-chain fatty acids, in particular butyrate, which is received and gained a lot of interest in this field, having anti-inflammatory properties. So we have been extremely happy to show that we have very few unique strains actually being able to produce butyrate, which can wonderfully be used in different therapeutic approaches. So now I would like to focus on one of the diseases that we have been focusing on, IBS, irritable bowel syndrome. As you may know, it is increased prevalence, approximately 10-15% of the inhabitants worldwide having IBS in one way or another. The classical symptoms are the abdominal pain, discomfort, constipation, diarrhea, bloating, and the symptoms are worsening by eating, which has been associated to the so-called leaky gut syndrome, increased intestinal permeability, and usually the patients just avoid the food they cannot eat, and usually these food and nutritions are the healthy one, including the fruits and the vegetables, etc. Patients in severe cases, they also develop depression and anxiety, and they cannot work, so it's a lot of burden and problem with them. This condition is also existing as a coexisting with different other conditions, including autoimmune diseases like MS, Sjogren's syndrome, and also other conditions like autism, fibromyalgia, and also chronic fatigue syndrome, which is in particular focus of our research and interest. I will get back shortly about that. So the patients, there is no good treatment available for this patient, as you know, and is very much self-managed condition, and they are seeking nutritional advice, etc. all the time. Considering IBS and having a kind of holistic view for producing and developing ImmuneBiotics for this approach, for sure we have been interested on the bacterial strains that have been associated to the dysbiosis observed in these patients, and which usually have been considered as a Clostridia, the strains of the Clostridia family. So having that in our mind, we have tested the inhibition impact of our strains on selected strains that has been known causing the problem. At the same time, the inflammation, there are studies showing that a great majority of the IBS patients do suffer from a low-grade inflammation. But this has not gained any much interest because inflammation has usually been considered important for IBD, but not for IBS. So we did consider that as important factor or mechanism to target, and for sure the strains, as you can see in the right panel, the first one screening after the properties, antibacterial properties suppressing the bacteria causing the problem, and they go to the next step of the screening, basically for checking for their potential to activate regulatory T cells, the suppressive immune cells, and that is a huge amount of work just testing these strains together in the combinations to achieve the consortium with the desired impact. And finally, after a lot of work, this is just a sample of what we have been doing. the impact on the barrier dysfunction, which has also been shown in IBS patients, indicating with the leaky gut syndrome, et cetera. Nobody has cared about that before. So we did have focus on that, having a consortium that could heal a leakage in the gut, which is in our major expertise to study and find a solution for. And of course, there are a tremendous amount of other parameters important to design the final product combination, which we keep it in-house, the knowledge and know-how. And all this work has resulted to GutMagnific, a product consisting of 5 lactobacilli strains, which has been tested, of course, in vivo. It is very important that that we test it in vivo scenario, both in animals and also clinical studies. That's our business model. Considering the animal models for IBS, the models available are predominantly stress-based models, basically targeting the intestinal muscle function, and there is a limited influence on the gut inflammation, intestinal permeability, and other important pathological parameters known in IBS patients. So based on our research on the EI model, the animal model for MS, and then the observations on the leaky gut and inflammation of the guts, we have chosen that model initially to test the impact of the product. And in this figure, and you will see the suppressive potential of GutMagnific in a protocol which is a very much tough therapeutic protocol, treating animals 2 weeks after the onset. And one important thing is that the efficacy of the product, of course, which is shown as a fresh frozen cells that we have been producing in the lab. But another approach I would like to emphasize here is the freeze-dried sample or product produced and developed by Sacco, which had the equal biological effect, therapeutic impact as the fresh frozen one. So this is another important thing, that although you may have the wonderful product working in animal models that most of the time you're producing in your own research lab, you should also consider the quality of this product while being produced in a large scale. That this should keep the same quality, and for that it's very important to choose the right actor, the right manufacturer, knowing what you're doing. And for this, we are very much proud and very happy for the wonderful work being performed by Sacco and the team. So the product, of course, in the initial form, while we were discussing the approach with the Sacco, has been tested in clinic initially as an enema, and the same protocol as fecal transplantation on patients with a severe IBS in different forms, diarrhea and constipation, as well as the patients suffering from Clostridium difficile infection. And we have and these studies have been initiated by the clinicians of interest, and they have shown wonderful results, and the patients have been followed 1 year after the treatment, no recurrence, no disease whatsoever, and they have all been released from the hospital. So it was their huge recommendation that we go on with the product, and that's exactly what we have done, and doing additional studies and tests in patients and also in animal models, finalizing the product. We have also done other important things. So the research is not really enough. So patent and protecting the product is very important. So to have the right consultancy agents knowing what they are doing. So we have been choosing and working with AVA Patent in Sweden, which have been wonderful work. And we have the pleasure to announce that our patent has previously been accepted in South Africa and other countries. But now, for a couple of months ago, we received information that has been approved in the US, which is quite happy news for us. Except the clinical efficacy, the regulation is also important, the regulatory requirements. Although we have done everything in our power to develop a product according to the pharmaceutical protocol, but the product we have decided to classify it as a nutritional supplement in order to market it and get around with the limitations available today. So production is very important. Choice of the manufacturer. After a month and almost a year of the investigation, we have chosen Sacco, and we have been extremely proud for the wonderful quality and the work they have done, the process development of all 5 strains, and that they have done successful large-scale production of all 5 that has been finalized in November last year, and the remained biological activity after freeze-drying process that was, exactly what we have been desiring. So we are very happy for that. And afterward, the product and powder has been shipped to another Italian company, Nutrilenia, which produced our capsules, and the packages been shipped to Sweden directly, ready for the market. And together with them, we have also been considering another cutting-edge ideas regarding the delivery solution, that we wanted the dosage of interest will be delivered safely to the right part of the gut, and for that we have chosen the capsules of the caps, vegetarian capsules protecting the bacteria from the stomach acid for the delayed release activities. So the product has been successfully launched on our web shop, GutMagnific.com, since October last year. And ever since, we have just received a tremendous interest for the product, for patients and individuals, consumers ordering it, coming back, telling us a lot of wonderful feedback about the health impact. We have been extremely happy, and reordering, recommending the product. So that's what so far it has been a wonderful experience, and even a new school and education for us, being transformed from the R&D company to the commercialization of this product. So more than hundreds of the individuals have been testing the product so far, and they have all shared wonderful stories about the impact on their IBS, both in the categories of diarrhea and constipation, leaky gut syndrome, and food sensitivity. Great majority with that issues, they just get back to us and told us that they could actually eat the food and nutrition they couldn't done for many years. And the impact on the SIBO, small intestinal bacterial overgrowth, which is a very tough problem to get rid of. You should need a broad spectrum antibiotics using for a long period of time, antibiotic-induced diarrhea, and also impact on the chronic fatigue in patients with IBS, myalgic encephalomyelitis, and MS, sharing the ideas about the impact on their gut, as well as the gaining more energy, and also minor improvement of the IBD patients. So for that, we are right now collecting these observations to make a careful study, and on these wonderful observations we have received so far of the great majority of the consumers. And this indicates that our product and research and idea, no matter how good you do your research in the lab, in the animal models, still no guarantee that works in human and in such a diverse population. So we are extremely happy and proud that so far we have received such a wonderful improvement, and we certainly believe that having the ImmuneBiotix and this consideration of looking at the condition as a holistic view, not just one simple problem to fix, we believe that has been the major background of our success. So we hope that this will continue. The sale has been increasing since October. We are extremely happy. We haven't, with a limited budget, a small company, we have, and especially we have been in the middle of the fundraising where the COVID scenario took place. So we have been extremely happy that the marketing has been very much thanks to the consumers, the power of the word and the recommendations. We are so happy for that, and we hope that this will continue. And we have also received tremendous interest from the consumers from US, where their relatives and friends have been purchasing the product in Europe, shipping over to US in different conditions. And thanks to all this observation, now we have received a extremely a huge order from one private person in US, which is a complete historical moment for our company. We will get back with more news shortly. So now we really focus on opening up to market the product in US. Now I will focus a little bit on our clinical investigation that we have recently started in collaboration with Karolinska. it's myalgic encephalomyelitis, or so-called post-viral fatigue syndrome, which is a disabling and extremely complex illness. The disease has also been called as a chronic fatigue syndrome, but now the scientist and also the clinician knows that their condition and problem is not only the fatigue. These patients are suffering from concentration, cognition problems. They have the chronic pain, light sensitivity, migraine. They have to be in the bed. They cannot move. They cannot do any sport or activity. They get worse by moving. So they should rely on the bed. They cannot move, being in a wheelchair or the bed long period of time, and mainly because of the light sensitivity with the sunglasses. So, and they have the eczema, allergy. So it's a tremendous burden. So there have been indications on the immune dysfunction. Natural killer cells have deficiency. They are very prone to the infections and the cancer. Mitochondrial dysfunction leading to the lack of the energy, brain inflammation, sleep disorder, and hormone disruption, etc., etc. But what has regained our interest has been the observation of the dysbiosis that recently has been very much focused on showing the dysbiosis of the gut microbiome in these patients, and the IBS, existence of IBS, and the great majority of these patients have had IBS before the disease or after. So the IBS is highly connected and associated to the disease, as well as the IBS-related symptoms like food sensitivity. So we have been extremely honored by invitation from Sweden's most important and only clinic focusing on diagnosis of ME/CFS in Stockholm's Rosendal and Karolinska Institute for a collaboration and testing the GutMagnific in patients because of the existence of the IBS. So after a couple of years of settlement and production of the capsules for the study, getting help from the SACO as well as the Neutrogena, during the time of preparation, we have also tested in several other patients suffering from ME and IBS. And in these pictures, you see Julia, which I had permission to show her picture. We're being in the bed for almost 7 years in a wheelchair, having IBS and SIBO. She has been improving the health with the impact on the gastro-related issues, as well as some other ME-related like brain fog, fatigue, and the pain, and starting to walk, and after 6 months starting to jog. So it's tremendous happiness for us actually having contact with these patients. But I would like to emphasize that our treatments or products are absolutely not a cure for the disease. So Julia and others still have ME, but we believe that these products or targeting the gut and gastrointestinal issues for them could improve their health and quality of life. So we give them a chance to at least can walk and enjoy the life, and at the same time perhaps more hope for the future, because the incidence of the self and suicide is extremely high in these patients, creating a lot of headlines in Sweden and other countries. So receiving grants from European Commission and other institutions in Sweden, we have moved further, and we have started our clinical study, a double-blind randomized placebo-controlled study to assess the effect of GutMagnific in patients with ME IBS. So the patients are supposed to have both conditions, and in 90 patients, so almost 100. So we are very happy getting permission for this, testing 2 different dosages of GutMagnific as well as a placebo. And there will be tremendous different readouts. So not only focusing on ME/CFS, but also on IBS, depression, anxiety, and also quality of life. We will collect fecal samples for different purposes, microbial analysis, as well as evaluation of the inflammatory markers and metabolomics. So finally, I would also like to present our companion probiotic approach, where we design and develop ImmuneBiotics to be combined with drugs to improve the efficacy and the safety, where a lot of drugs and immunotherapies, as you may know, have as a side effect a lot of gastrointestinal problems, causing gastrointestinal problems in patients. So improving the efficacy and the safety of these drugs can advance the products as a better and safer drugs for reaching more patients, better position of the drug, extending the patent and the protection for the pharmaceutical companies owning the products, and of course being offered as an alternative for patients in risk. So this has been very much welcome for a pharmaceutical company, and we are very happy establishing collaborations. And right now our focus is autoimmunity, and in particular a product for multiple sclerosis, and then testing the impact on a very selected drugs with issues. So a little bit on the pipeline. So the GutMagnific has been going very far from research now into the market, and evaluation and clinical studies ongoing, additional. And our companion probiotics, the first product is focused on MS, and the upcoming products will be having focus on the IBD. So a little bit of current situation and challenges. So although the COVID pandemic have been giving a major impact on all of us, and not only on the business, but also the co-workers' health and quarantine. So basically the planning is day to day. There is an increased interest for our product. The clinical study we have been setting up in an approach being very much controlled by distance, because even the ME clinic and rehab clinic in Stockholm has been working under pressure during this pandemic. So we have taken more responsibility communicating with the patients. And of course the delays in a Series A funding where we have been in the middle of. But at the same time, we have received the increasing reports from consumers on the significant impact of GutMagnific on the respiratory infections. So patients with the family members suffering from influenza and ordinary cold, they have reported that they have been suffering less often and less severe, which is very interesting for us, actually. And also some COVID patients in US have been reporting basically in their daily report showing after the diagnosis of COVID, getting access to the GutMagnific, how they had a very quick recovery of the disease and the gastro issues, which is very much correlated to the COVID-19 pathogenesis. So we are, of course, very happy for that. It's a very good surprise for us, has not been planned. And now we have set up a collaboration with the research group, respiratory immunopharmacology group at Lund University, to further evaluate the antiviral activities of GutMagnific. And the increasing sale is continuing, and we hope that we can be healthy enough that can manage this and finding good partners, collaborations, and reaching out to more patients, more consumers, both in Europe and opening up in US, and also looking very much forward to collaborating with the business-to-business partnerships. This is my team. It's a small team, but at the same time, we have all the knowledge and experiences necessary within that, skills. So Karin Larsson, our business manager, CBO, is a wonderful person, having also a PhD, having insight both in science and the business. Menos Nouri, the co-founder, she has very much been managing our bacterial library and very much as an immunologist, but also aware about the probiotic research. Greg Batchelor, our chairman strategic advisor, has been involved in many successful companies in Lund, a very famous guy in Lund, actually, but also living in Belgium and China. So we are very happy and honored to having him on board, and we are surrounded by a wonderful scientific advisors, initiating ideas for product development, as has been for GutMagnific, and also interest for evaluating the product in academic kind of collaborations. So we are extremely proud and happy for this faith and trust, and we hope that we can do more. So I would like to thank you so much for your attention, and you're very much welcome to reach out to us by this email and phone numbers. And with that, I open up for questions, and we'll be very happy to answer your questions. I will.
So thank you, Sharon, for this nice presentation.
Thank you.